faster, and better your success to IND and BLA at Samsung Biologics. Samsung Biologics is your most qualified CDO, CMO, and CRO partner. Starting from cell line development, process development, DS manufacturing, DP manufacturing, DS and DP stability study, and regulatory filing support, all is completed in 10.5 months. For all different types of biologics product, we help our clients to bring their ideas into reality with our development strategy to maximize client satisfaction. Our competitive timeline allows us to deliver cell line development to DS production within 9 months to DP production in 10.5 months. In addition, Samsung Biologics now offer proprietary cell line, providing our clients another option. Our new CRO business allows us to perform more integrated services at one site. Our contract research organization now supports biosafety testing and standalone contract QC services. With 30,000 liters Plant 1, 154,000 liters Plant 2, and 180,000 liters Plant 3 capacity, our clients can benefit from maximum manufacturing flexibility. We are the largest CDMO with the latest technology such as N-1 perfusion and Beacon platform. Our excellent quality operation is also proven with 51 approvals from global regulatory authorities. As your most qualified, world-leading CDO, CMO, and CRO partner, Samsung Biologics delivers your success to IND and BLA faster and better with maximum flexibility. We promise the best.
Hi, everyone. My name is John Gill. I'm the Director of Cell Line Development at Samsung Biologics, and I thank you for joining me as I speak to getting to your IND faster and how we in Cell Line Development have developed a platform for accelerated and high-performance cell line development that allow you to get from start to finish in 10.5 months. Again, my name is John Gill. With the nature of the presentation being in web format and me not being there for questions, I'm going to keep the presentation relatively informal and really encourage you to reach out to me or our business development team for any questions that you may have regarding any of the content or services that we provide. I've been the part leader or cell line Director of Cell Line here for 1 year time. And prior to that, spent a number or the bulk of my career at Cherry Pharmaceuticals in upstream cell line development and discovery roles. We're going to talk about a couple of things. The first is really just the CMC activities needed for your IND and the fast timeline. And again, the enabling of that timeline through the accelerated and cell line development platform that has been established here. First, we'll dive into the CMC activities. Your success to IND, faster and better here at Samsung. We're reviewing the development pathway from the gene to the IND. We start here at Samsung Biologics with receipt of your sequence for your gene of interest. We do all of the vector construction externally and start our timeline really at the transfection stage. The cell line development is then initiated. We contract the MCB banking and characterization, but simultaneously transferring any of the research cell banks, so the pre-master seed, to our process development organization, where we initiate the upstream, downstream formulation and analytical method development activities, which facilitate the tox or non-GMP DSDP production for your IND stability start. Still internally, offering both the GMP, DS, and DP manufacturing and stability, and culminating in the IND filing support that we offer here at Samsung Biologics. The overall timeline from CLD to DP manufacturing is 10.5 months. I say that for the presentation only, because as you come to us for any of our technical services, it's highly flexible or modified to your particular needs. The way that we structure both of the activities and the LOIs is really just where you want to come in and out of your process development or CMC activities. For us, as I said before, the vector construction happens externally, and there's a 3-month timeline that we'll go over for the cell line development. As we initiate the cell line stability stage, we move through the process development activities. This timeline is considered for IgG1, IgG4 simple mAb molecules, where we can take a platform approach to the process development activities. To manufacturing, leading to, again, all of the documentation support and filing needs to get your IND out of the door and to your regulatory agencies. So the rest of the presentation is really regarding how we enable the start of your CMC activities from a cell line development perspective. And that's accelerated through 2 main platforms. One, the single cell isolation stage, utilizing the technology from Berkeley Lights, the Beacon. So through optofluidic technology, we're able to screen up to 6,800 clones in the middle of our CLD platform. That gives us a much higher accuracy in correlation of ranking for the eventual productivity and cell growth that you would see in your small scale bioreactors. Additionally, this provides you with the documentation support that you would need for the monoclonality, with a visual record included in the cell line development reports and documentation of greater than 99% of assurance. The cell line development is completed with a 3-month timeline from transfection to the RCB, with high titers in the 5 gram per liter range. While we don't guarantee any result on titer, it's really quite robust from a monoclonal antibody standpoint. So from a majority, I would say all of the programs that we've had to date, that 5 is right in the middle to lower range of the titers that we have been sending the clones out the door to our upstream colleagues. The initial platform itself is through electroporation, selection in mini pools, ranking in deep well plates for titer and product quality, single cell isolation on the Beacon, and then another ranking or ranking stages in the scale up of 24 to 48 clones through deep well, amber, 15 manufacturability assessment, leading to your pre-master seed or your RCBs. So how we enable that is through 2 host cell lines. The primary driver at the moment is through Chosen, the SAFC cell line. But however, we have also developed an internally developed program with this also a CHO K1 GS knockout cell line developed here at Samsung. We licensed the CHO K1 cell line from ATCC, and then through the process of TALENT mRNA knockout, isolated a number of individual clones through multiple rounds of screening, 2 rounds of phenotype screening, just growth promotion plus and minus glutamine, sandwiched around a sequencing step to assure that we have true knockout of the glutamine synthetase gene. From this point forward, we took all of the positive clones that through a media adaptation stage. And while I say the word media adaptation, it was really just serial passaging for a little over a month's time, just to monitor the cell growth and viability over that period, to allow us to select in the top 5 of these potential host cell lines for performance testing. What you can see from this period of the screening is very robust and repeatable growth as we transitioned the cells from the initial CD-CHO media to the Excel CD fusion from Merck. The doubling time for the 5 host clones that we selected for this ranges from 18 to 21 hour doubling time. And then at the end of the study for passaging, we went through just a simple 6-day batch culture, just looking at the peak VCD at the end of that stage, where compared to the control, where we were a little over 4 million cells, we almost saw a twofold increase in your VCD at these early stages, which means you're getting closer to the heavy lifting of your fed batch process when you're getting to your peak viable cell densities. So what we did with these is created a research cell bank for each of these 5 clones and then selected them for a performance test. What that looked like is a full cell line development workflow, expressing here an antibody, an IgG1, with all 5 of those hosts, doing the mini pool selection, plating in 10 96-well plates, going through the stable pool selection, screening by octet. The top 10% of those pools were carried through the ranking and scale-up, where we looked at the 24-hour QP, leading to 2 fed batch processes, one 11-day in the deep well plates, culminating in a 14 fed batch production study in the shake flasks. At this stage in our development, we didn't do any particular development on the expression vector. It's just something that I want to point out now because later in the talk, I'll explain some of the improvements we've made to the vector itself. As I jump through our performance testing, we're looking here at the culmination of the stable pool screening. So we took the top 5 pools from each of those CHO controls and plotted the 14-day fed batch processes against our control. So the CHO control here for this IgG1 was 2.9 gram per liter for the stable pool. And looking at clone 48 and clone 56, the top 5, or actually the top expressing clone or stable pool was 3.6 gram per liter for the clones from 48, so a 24% increase over our control. But then looking at the far right, clone 56 derived clones, we hit 5.7 gram per liter from our stable pool, which was quite exciting because what it allows us to do is initiate our upstream process development a little bit earlier in the CMC timeline to generate material for any of your formulation development or even your upstream and downstream screening. My preference is to get some of these stable pools into the upstream development hands so that they can do some preliminary media and feed evaluations so that when we do the final screening for the candidate clones, we have a little bit more catered or custom media that's more relevant to the clones that we're selecting and transitioning as candidates to the master cell bank and any of the upstream process development later on. But again, we were quite happy with the titer, 5.7 gram per liter at the stable pool stage. So we went through the single cell cloning. For this, the performance test, we were still in the pre-optimized or pre-institution of the beacon technology. So the single cell cloning was done by Cytina with imaging on the cell metric by Celentum. And I'll talk a little bit more on the clones on the next slide. Really, this data that we'll show here is what I think truly leads to the performance or benefit of the cell line that has been developed here. If you look on the far right for clone 56, this is the VCD and viability over that 14-day fed batch. And you can compare that to the CHO control on the far left. What you see on the right with the clone 56 is not the typical 14-day fed batch where you see a peak VCD on day 7 through 9 with the drop in viability and VCD for that remaining week of the manufacturing process, which is quite significant in that it can lead to higher titers, but also better product quality from end to end. As mentioned earlier, we went through the single cell cloning stage, isolated those, scaled them up for another 14-day fed batch processing. The media here was the Excel Advanced Fed Batch with feed from High Clone Cell Boost 7A and 7B with a working volume of 50 mils. The end story here, the clones derived from the control, while we did see an improvement of from 2.9 to 3.4 gram per liter, our stable pool of 5.7 still had enough diversity within that population that we were able to isolate a clone at 7.6 gram per liter with this new host developed internally. The next stage of optimization was really just looking at some seed and feed screening, where we plotted that particular clone that was expressing 7.6 gram per liter over a 20-day fed batch process. Not that we would recommend this for your manufacturing, but we wanted to really look at the robustness of the host and see if we were at the edge of the failure at the 14-day stage. What you can see here at the end of 20 days, we're still maintaining viability at the 90% or greater range, which again then shows that we're hitting peak VCD between days 5 and 7 and maintaining that all the way through your 20-day fed batch processing. What that ended up yielding is 11.6 gram per liter at the end of 20 days, but we're more scalable, reproducible, or targeted manufacturing with a 14-day fed batch. We're close to that 10 gram per liter range, again, without an optimized vector system. I'm going to go over this slide very quickly just to point out that we looked at a number of different promoter and intron combinations, yielding a twofold increase in the overall GFP expression. So we took that initial vector that was used for the performance testing, cloned out the monoclonal, and threw GFP in there to allow us for some quick screening of these combinations. And you can see that, initially that we did see quite a bit of promise. For you CLD experts, you'll know that GFP doesn't always directly correlate to any actual secretion of your targeted proteins. So the next stage was, again, then swapping out the GFP and looking at monoclonal and F molecules. And what we're really happy to see is that twofold increase that we saw with the GFP truly held for the IgG1 and FC fusion molecules. So we did again see from the control a twofold improvement with the PSBL9A expression vector that was established here, and a similar increase with the FC fusion. So to wrap up, the competitive fast timeline, again, is what we showed here is 3 months for CLD and 10.5 months for the overall process. But again, it's customizable to your molecule and to your particular needs as a seamless one-stop shop service. In that, you're getting excellent quality and outstanding capability. We have excellent scientists here and good technology, yielding to high titers with very good product quality. There are over 2,600 scientists or employees at Samsung Biologics. Over 1/3 of those are in the quality organization. So that's led to this point over 51 global regulatory approvals. One of the keys that most clients are interested in is on the pricing. So the Samsung Biologics pricing is very competitive. So there's no binding structure and no royalty payment for the cell line. And again, we really stress the competitive pricing with the best quality, leading to overall client satisfaction. We have dedicated program managers. So for each of these particular programs that we'd work with our clients, you're talking to the head of CLD, the SME, that's the bench scientist or bench scientists that are working on your program, and 2 project managers, a senior and junior project manager, to make sure that the communication is open, transparent, and ongoing through the entire collaboration. The last update is Samsung Biologics, US R&D Center opening sometime in 2020, more related to the COVID situation than anything else, but we're excited to bring our capabilities and our expertise to the US. We'll have more on that in the news very shortly. With that, as your most qualified world-leading CDO, CMO, CRO partner, Samsung Biologics delivers your success to IND and BLA faster and better with maximum flexibility. We truly promise you the best, and I look forward to hearing from you either through email, phone calls, LinkedIn. Ask away. Thank you.